But some of this actually goes back to when Anderson was a student, and he probably remembers some of the people who contributed. So this is work that's gone on for a long time in the group. So I want to give you a bit of a history of things. So this is the way I think about it. You know, this is the way I learned about electronics when I was young. This is a vacuum tube. How many people have ever seen a vacuum tube? I don't really believe you, but okay. You're too old, too young. But this is what I studied when I studied electronics. Of course, that went to transistors. This is what I learned in early university. That's gone to integrated circuits. Now you have microprocessors, and now you have these massive microprocessors, right, by NVIDIA. So everything has gotten smaller and smaller and smaller. Well, the same thing has happened with fluids. So this is the way we normally do things in a lab with a pipette, and we use a milliliter of fluids. But this is changed and evolved so that we now use robots to do this. This is still ancient generation, maybe 96 wells. Now there's 1596 wells, but all done just by using a robot and a pipette. But just as electronics have evolved, so too have microfluidics. And they've gone now to small chips that don't control electrons, don't control the flow of current, but control the flow of fluids. And this is something that's evolved over the last 10 or 15 years, 20 years. I think that maybe one of the biggest innovations in this occurred when one of my former colleagues at Harvard, George Weitz, found or discovered a very simple, or popularized actually, a very simple way of making chips like this. Normally, it was done by using the same technology that you use for making microchips for electronics that maybe 20 or 30 years ago. Quite old technology. But the problem with that is that it took maybe a month to produce a device. With George's innovation, which is soft lithography, which I'm not going to really describe, you could make these devices now in a day or two. And this really led to a big revolution in fluid control. This particular chip is actually a fairly old one, but it's something that was designed by one of the real pioneers, Steve Quake. And now microfluidic devices have many, many uses. And I'm going to actually try in the 4 hours, in the hour I have to describe two generic kinds of uses. Most of the time, I'm going to not focus on the major use of microfluidics, but I'll try and describe some of that afterwards. And we do really a lot of work on trying to use what's now called lab on a chip. By the way, lab on a chip, people like Anderson really love this idea because he has this new faculty member. Anderson says, "Oh, what do you need?" He says, "I need a lab space." He says, "No, you need a lab on a chip." And he gives them just a little chip to do it instead of the lab space. You can just shrink everything down. So people like Anderson who have to deal with important things like space and things like that love this idea of lab on a chip. And that's really a major application of microfluidics, which I will describe in the last few minutes of the talk. But what I really want to discuss is making materials with microfluidics. This is not something that is done as commonly, but it's something that in my group we've spent a lot of time trying to understand, could we make really new kinds of materials? Could we do something different with microfluidics? And I'd like to show you, I'd like to convince you that we can make really interesting kinds of materials. They have a lot of practical uses, but you should be a little bit cautious because what I'm going to show you is we're going to make things with microfluidics, very small scales. And you might ask, and you should ask at the end, that's very, very nice, or I hope I can convince you it's very nice, but can you make it at a scale where it's actually useful? And I'd like to try and also convince you that you can do that as well. So what we've pioneered and really worked hard on is just making drops of one fluid in a second. That's something that's very simple. It's not the traditional use of microfluidics, but it's something that we worked hard to make into something fairly simple. And the rationale for that is that you can make a drop very, very uniformly. And the beauty of a drop of one fluid and a second fluid is a surface tension. Surface tension makes the drop into a perfect sphere. It really keeps it spherical. But we can use that drop to build other structures around it. And that's what I'm going to focus on today. So we use the drop as a template, and we fabricate new kinds of materials with microfluidics. Now, we were doing this many years ago. We were across the street from George, who'd done all this beautiful work making devices very simple, but we wanted to make materials rather than to try and use lab-on-a-chip technology. And then you want to use a variety of different solvents. You don't want to use the simple solvents that people normally do with soft lithography. So we needed to develop something that was really simple, really reproducible, very easy to make, but was made with something more robust for different kinds of solvents. And the way to do that, of course, is to use glass, but we don't want to use the traditional glass that's used for lab-on-a-chip technologies, simply because making a chip like that in those days certainly would take several months, or at least a month. So what we did was we developed something we call capillary microfluidics. It's really very, very simple. That's the beauty of it. We take capillary tubes and we go across the street to the biology department where they do patch clamping, and we ask them, can you please teach us how to make a sharp point out of these tubes? So these are capillary tubes. They're each 1 millimeter in outer diameter, 0.5 millimeter in inner diameter, absolutely well defined, both the inside and the outside. And you go across the street and your friends in the biology department teach you that the capillary tube is made of glass, so you can heat it up in the middle and pull it apart. And you do this just with a machine that does this very reproducibly. And then you can make it into this very, very sharp point like this. This is seen with just a camera. I'm going to show you pictures with a microscope. But now you have to make a device, so you have to line things together. I guess I'm missing some slides. Do I have them? No, sorry. I'm missing some slides here. Oh, sorry. Not used to this machine. Sorry. What we do, the slide that I'm missing is that to align the device, what we do is we have a square capillary. A square capillary, we're putting a round capillary into a square capillary. The square capillary has exactly the same inner dimension as the cylindrical capillary here. And you can slide the cylindrical capillary into the square capillary, and you can align it perfectly. And so I'm going to show you ways of doing this that you can align cylindrical capillaries in a very simple way. And it's all held together. I told you it has to be something very simple, and so we like the simplest material possible to hold it together, just 5-minute epoxy. And Anderson, when he was a graduate student, could make these kind of devices and much fancier devices in roughly half an hour, very, very simply. Something that you could learn. It's not complicated. We still make them in our lab. It's not complicated, and you can make rather sophisticated devices very quickly. And that's important because what you don't want to spend your time making devices. You want to spend your time making materials. You want to spend your time trying to do things with materials, not making the devices. This is what it looks like in the lab. You have to put it all together. You need to look at a microscope because it's small, so you have to connect it. We use just syringe pumps. Everything really very, very simple for this laboratory scale experiment. And with this, we can make drops. We just simply push one fluid through the inner capillary, the other through the square capillary on the outside. And you can see that we can make drops, and the drops really are very, very uniform. They're going one by one by one. You can't tell the difference in their size. They're really very, very uniform in size. And this should remind you of something. This should remind you drip, 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 drip. So we call this dripping. But if we increase the flow rate, then we get something that it forms a jet. I'm going to try and hold this laser pointer very steadily. And you see sometimes the drops form where the laser pointer is, but not always. But they always form drops. And this is absolutely intrinsic when you have a jet of fluid, when you have a thin stream of fluid, it's unstable against forming drops. So any kind of fluctuation will ultimately lead it to form a drop, and you get drops in the end. So you get all these drops of different sizes. So this is the geometry that we use. This is the co-flow geometry. We have the two fluids. I should have shown this earlier. We have one fluid in the middle, the other two fluids in the four interstices here, and you make drops, and they're flowing in the same direction. But you can do a different thing. This actually, what happened here was an interesting thing. A new graduate student came, and he tried to do something, and he went away, and he connected it all together, but he connected it the wrong way. He made a mistake. And as always with good graduate students, if you make a mistake, you learn something new. And he realized that you could get the same effect, but now, instead of having the two fluids flowing in the same direction, you have them flowing in the opposite direction. This one still goes through this narrow orifice, but now the other fluid is coming around here and focusing it through this. So we call this a flow focusing geometry. And what's nice about this is because it's flow focuses, this stream is a little bit narrower than the orifice. It still works. Here's an example. You can see the drops are being made here. This is the fluid coming from over here, and this fluid is coming from way around here. But it's going through, and you're forming drops. This is the dripping regime. You can go faster, and you form a jetting regime. You can barely see the glass capillary in this case because the index of refraction of the fluid is the same as the glass. But you can see there's a little piece of dust here that was a remnant, so you know where the orifice is. It works exactly the same way. But what's interesting about this now is I told you because we're using these square capillaries to align these cylindrical capillaries, we can align cylinders very precisely. And so if we look at this, we have these fluids, but we have a lot of space right here. So let's just add a second capillary tube that looks like this. And now we have three fluids. We have the co-flow geometry here, one fluid and a second fluid, all flow focusing with the third fluid. So now we have three fluids. So here's I should have shown you this before. This is the square capillary. We slide the cylindrical capillary into the second one. We slide the second capillary in here. And if we look in the microscope, you can see that we can really precisely align them. And we can do this very, very precisely because it's so easy to align using just the symmetry of the geometry, the symmetry of the square capillaries and the cylindrical capillaries. You just put them up against one another, and you can perfectly align them like this. And so this is what it looks like. Here's one capillary, a second capillary, and a third capillary. And now you can see we're making drops, but we're making drops inside of drops. Maybe it's better to look at this one where we've changed the flow rates. And here you can see a large drop with a small drop on the inside. Here's a drop with a larger inner drop. This is actually, you might say, well, this is only a single drop, but if you look carefully, there's one fluid, two fluids, and three fluids. And if you look very carefully here, you see there's a very thin shell. And this one, it looks like they're flowing the wrong way, but that's just a stroboscopic effect. You're making them, and you're making these multiple emulsions. So these multiple emulsions are already very, very interesting materials. Let me show you a simple example of what you can do with them. So what I've showed you is you can make one drop inside of a second drop, a double emulsion. This is what I'm showing right here, a double emulsion. One drop inside a second drop. Well, let's imagine that the outer fluid is water, the inner fluid is water, but this shell, the middle phase, the shell is some solvent in which I put amphiphilic molecules, molecules that will go to the interface of the solvent and the water, the solvent and the water. So they will coat both interfaces. And I can use surfactants, I could use phospholipids, I could use diblock copolymers, I can use any kind of amphiphilic molecule. But then if this solvent is slightly volatile and slightly miscible in the water, this solvent can be essentially evaporated and removed. And you can end up with a very thin shell, which is just made up of the two layers of the amphiphils. Maybe phospholipids. If they're lipids, you have a vesicle. If they're diblock copolymers, you have a polymerisome. So you have just a bilayer structure. You evaporate everything else. But what's interesting about this, actually vesicles like this have been used for a long time to try and deliver drugs. The problem with them is you make these bilayer structures, and then you have to fill them. In this case, the two fluids are completely separate. They're separate flows. So this fluid can be completely different than this, or can contain something that's completely different. So this is a way of encapsulating something very, very efficiently without any losses. This works quite well. Here's an example. In the days that we started this, we were really most interested in polymerisomes. They had just been developed at University of Pennsylvania, where I had just been. Dennis Disher and Dan Hammer had done a lot of work with polymerisomes. I was colleagues of theirs at the time, and we were very interested in learning how to make them. You could make them with these biocompatible polymers, PEG and PLA. It's very difficult to actually self-assemble structures like this, but with this directed assembly like this, it's very easy to make these structures. And you can see here the bilayer structures, the polymerisome structures. You can see there's a little excess amount of diblock here always, because you don't want to put enough material so you can completely coat each interface. So you put a little extra, and you get this little ball of excess material. In this case, we encapsulated a fluorescent dye, and we can come back months later and it's still there. It's a very, very effective way of encapsulating something. It's interesting to watch what happens when the solvent evaporates. Here's some images of this. You can see what's happening. This is the bilayer structure, and this is the evaporating solvent. Here's another example. This is the bilayer structure. Here's the evaporating solvent. And look, there's a contact angle here. If there's a contact angle, there must be some adhesion between the two structures. So there has to be something that is adherent between these two structures. If that's the case, you can, first of all, control the contact angle by using different solvents. You can adjust it so you can make it as large as 90 degrees. And you find that when you make them as large as you can, then the stability of the structures increases, it improves. So what's happening? Well, if it's two structures, two layers of amphiphils that are adhering to one another, then it's really unlikely that that's a fluid membrane. It should be something, if it's adherent, it should be something that's solid. So how can I prove to you that it's a solid? Well, one way is to take a structure like this and crush it, like take a sledgehammer and just crush it and see what happens. Of course, using a sledgehammer is not so good in a fluid. So how can I do that? Well, I can use osmotic pressure. I can put an osmotic pressure and crush these things. And look what happens when I do that. I put an osmotic pressure, and when they crush, they don't flow. They buckle. And buckling, something that buckles, has to be a solid. So this shows immediately that the shells are actually solid. They're a weak solid. And you can measure the elastic constant just by measuring how much buckling occurs as a function of the pressure that you're applying. Notice the pressures are in tens of atmospheres. That's typical of osmotic pressures, tens of atmospheres. So it's quite large pressures. And you can see that these buckles, and my theoretical colleague David Nelson, we were able to calculate what the elastic constants of the shell is just by measuring the shape of the buckling that you see. It's actually interesting to watch what happens when you buckle. Here I'm going to show a series of images taken over this course of several minutes. You're allowing these things to be compressed by osmotic pressure. And here's what looks, after 13 minutes, it's completely compressed. So now just think of what's inside of it. Anything inside except maybe a few massive biomolecules. Anything else has been squeezed out. So not only have I showed you how to compress these things, not only have I showed you how to encapsulate something, but I just showed you how to release it as well. I just put a large pressure and squeeze it out. I can do other things. I can take advantage of the difference in osmotic pressure, and there is, because there's some small molecules, for example, the fluorescent dye, in this case, the ions, the dye molecules themselves, form their own osmotic pressure, an internal osmotic pressure. If I put it in a very low osmotic pressure medium, like put it in pure water, then this osmotic pressure is much higher, and this is actually what happened. We did this, and a little hole developed, and we come back half an hour later. I can't show you a fluorescent image anymore because the dye has all escaped and diffused away, and I have to show you a bright field image. And it's actually interesting to watch what happens when that occurs. Here's one of these shells, and we're putting it into a low osmotic pressure medium, and watch. First you see that it actually grows a bit, and then boom, a hole develops and it starts to shrink. Look at this one. It grows, so it always grows a bit, then the water is rushing in, then the hole develops, and this one's completely gone after a few minutes. You can do other things. You don't have to evaporate the solvent. Here's a case where we just use UV and polymerize the solvent. Now to release things, you actually have to literally use a sledgehammer. You have to squeeze it between your hands, and you have to crush it. It looks like this. You can do many other things. Here's a case where we did what we called melt emulsification. That is, we used a relatively low melting temperature oil. This is triglycerides. It happens to melt around body temperature. It's something, for example, that's often used in chocolates. And we then heat it up. We have a fluid. We make the emulsions at high temperature, and then we cool them down, and we have these solid-like structures. And these, to melt, to release them, now we can just use temperature. So here's a case where here we have some colloidal particles, here we have some dye, and I'll show you what happens as we heat it up. You see that the shell completely melts. This already has interesting implications that we were thinking about. For example, if you want to deliver two types of drugs, and this is actually very common where you want to deliver drugs synergistically, they work synergistically. You want to do several, let's say just two drugs at the same time. You could encapsulate, for example, one drug on the inside, which is the hydrophilic region, one drug in the shell, which is hydrophobic region, and here we're just using dyes as a mimic of the drugs, but we could use drugs like this. And then to release them, we just have to heat them up, and they'll release exactly at the same point. So here's the two different dyes. You can see when they're released, they released exactly at the same point. And this was actually very useful. We used it just in some cancer cell killing assays, just where there are two drugs that work synergistically very well to kill the cells. And we showed that that was a very effective way of delivering drugs. Jörg Werner was much more interested in using different kinds of polymers. So he used polymethacrylate anhydride. This is a polymer that swells by changing the pH. So we made a shell of these things, and by increasing the pH from 4 to 7, it would swell. Here's a movie of this thing. You can see that it gets larger. It buckles first, but then it gets larger. You decrease the pH, the shells get smaller. So you can make them larger and smaller. And what that means is you can also change the pore size. So you can change the permeability of the shell just by changing the pH. What that means is you can control release just by having something that will release when the pores are large. When it's swollen, the pores get larger. But when it shrinks, it doesn't release, so you can control the release. You can control it over time. You can just by adjusting the pH. You can do a variety of different things where you can, instead of doing pH, you can have something that will degrade in time. Here's where he's controlled things where he can control the release essentially instantaneously, or something that will release over several days. Again, just by controlling the exact of the nature of the chemistry and how it evolves in time, you can control the type of release that you can get. Well, there's other ways that we can make this. We had a visitor who decided he'd try and do something a little bit different by using the fact that you could align the capillaries so well. So what he did was, instead of having a flow and a co-flow all at the same point, he separated them. So he had 3 capillaries, one capillary making drops through a co-flow mechanism into a second capillary and producing drops this way. And the nice thing about this, because now it's all in the co-flow geometry, you can very precisely control each of the fluids. And so now you can see, look, you're making drops. Here the drops are coming in. There's 2. A third one comes and it snaps off. I've watched this many times. It snaps off right now. You have much higher control, so you can make what I call designer emulsions. Now you're not just making one drop inside of another, but you can make any number of drops. So I ask you, how many drops do you want? You say, oh, I want 5. Well, I say, how big do you want them? You see, you can control the relative size, you control the relative number. These are all just sample drops of many that are exactly the same. So you really have control of this. But he showed me this, the visitor showed me this. I said, well, that's great, but we've done this. What else can you do? Well, he said, look, I have one drop maker, I have another drop maker. What about here? Let's add a third drop maker. So now you have drops inside of drops inside of drops. And you can do that very precisely. And again, you have perfect control. So these really are designer emulsions. And he showed that to me. I said, that's beautiful. But why would anybody care about that? Show me why it's useful. So he made this structure. Here's a drop inside of a shell. This is made by using the fact that you can control this. And he made this into a hydrogel. And he put this, these are water drops in oil in a continuous phase of oil. But inside these water drops, he put something that's a very delicate molecule, a peptide, that you don't want to be denatured. Outside, it's essentially a cream, and there's a lot of surfactant. And if you put it there, it would denature the peptide. Here, you can protect it very nicely, and this is perfectly protected. But this is thermosensitive. So you heat this up, and it shrinks. The gel shrinks. It tries to compress. It can't compress the inside, so eventually it just tears itself apart and releases this. So this is now really a way of absolutely, of very, very robustly encapsulating something and very controllably releasing something. And if you think very generically about encapsulation and release, it's very easy to make something that very robustly encapsulates something, but it's actually difficult to control its release. It's also easy to make something that you can release very easily, but then typically the encapsulation is not so robust. Because we have this multiple levels of fluid control, we can do something, we can do both of them. We can both encapsulate something and release it with a very high degree of control. So there's other things that we can do. I won't go through the details, but we can take advantage of the wettability of the glass, and we can make very thin shells. When we do that, we can make structures, so we can make vesicle structures. It's actually difficult to make vesicle structures with that contact angle because the contact angle tends to break the vesicle structure. But if you have a very thin shell, you don't have a contact angle. Instead, you have a little contact line, and that's not as difficult. And now you can see you can make these very large vesicle structures. They're so large that thermal energy causes them to fluctuate. Here you can see the small contact angle of the final fluid disappearing, and you can see these fluctuations. And so Laura Arriaga used this to study the behavior of phospholipids on the interface and how they undergo different kinds of phase separation just on the interface of these structures. Here's one where she used 2 different dyes and show how you can see phase separation of the phospholipids on the structure. But then we asked ourselves, could we make something even more complicated, something more sophisticated in terms of the structure? So this is this traditional way, and as I say, vesicles like this, or small vesicles called liposomes, have been used for years, and they're always made by using the same type of phospholipid on the inside and the outside. But we asked ourselves, could we make something where we had different phospholipids on the inside and the outside? So how could we do something more sophisticated? Well, we can make these multiple emulsions that I showed you. So instead of just using one shell, we can use 2 shells, 2 solvents, one with one type of phospholipid, the other with a different type of phospholipid, and thereby make asymmetric structures. And to test whether there is asymmetric, we can just use a dye binding assay. We can put something that will bind a dye. We can put it on the inside or the outside, and then we expose it to the dye. The dye will bind if it's on the outside, won't bind on the inside. So you'll see fluorescence here and not here. And here's an example where you can see that's exactly what happens. This structure, the binding of the dye was on the outside. You can see a lot of dye binds to it. In this case, it was on the inside. There's a little bit of dye that binds to it. That means that some of the molecules on the inside ended up on the outside. It's not 100% asymmetric, but it's pretty good. We estimate it's about 80% asymmetric. And you can do other things. So Tina Wong was interested in trying different things. Why just two types of phospholipids? What about a phospholipid and a diblock copolymer? This will give you much more flexibility if you want to do some kind of delivery. You can use something that's very biocompatible, that's the phospholipids, something that's very robust, that's the diblock copolymers. And when she did this, she could make these kinds of structures. You can see these looking at different types of behavior. And by controlling the relative amounts, which was in the inside and which was the outside, she was able to show that you could get very different types of structures. And this is just measuring a stress-strain relationship. This is the normal phospholipid. You can see it takes very little stress to cause it to strain. What she's doing is she's using a micropipette. She's sucking the structure into a micropipette, using different pressures to suck it in, measuring what the displacement is. And by changing the asymmetry, this is the phospholipids, she puts a little bit of diblock in, it gets a little stiffer. Put a lot more diblock in, you start getting something that's really quite stiff. And she can now look at the different behaviors where she puts different amounts of the material, different types on the inside, on the outside, and just measure the consequences for the mechanics. Kevin Janke, actually working with ABIC, now took a slightly different approach. He wanted to make much smaller structures, but again, trying to make these asymmetric vesicles. He used a slightly different approach. It's not the normal capillary microfluidics. It's a different kind of microfluidics. It's just basically using an extruder, which is, if you want, it's just a very, very thin, thin membrane with many, many highly controlled holes. So it's like the capillary. You're squeezing the drops through. But then he used a trick that was actually developed years ago in our lab, but never really followed up by us. But he's followed it up where he makes a water emulsion with one type of lipid on the inside in an oil, and then he passes it through a second monolayer of a second type of phospholipid. He passes it through, and he can make these asymmetric structures. He finds that he can make them very asymmetric. Again, he measures the asymmetry with the dye test, and he finds it about 75–80%. He can make them very, very small, which is what he's interested in because he wants to use these for drug delivery. So now he can make structures that are asymmetric, that he can fill with whatever he wants, and he can ask, are these good for drug delivery? The motivation, of course, for this was really thinking about the importance of using phospholipids for delivering drugs, which we learned during the pandemic. For those of you who got a vaccine, an mRNA vaccine, either the Pfizer or the Moderna, in Boston, of course, we all got the Moderna one because Moderna is in Boston, but those absolutely depended on the delivery using phospholipids. It's a fantastic technology that they use, but it's not really great for delivering all kinds of other modern drugs. This is a structure that would be much more suitable for many different types of drugs. And so Kevin showed that by varying the different types of asymmetry, you can put the charge positive or negative or uncharged on the inside, the outside. You could vary the charge. You could put the charge on the inside, the charge on the outside. You can put positive charges on the outside so it goes to cells more easily, negative charges so it controls what the type of molecules are. You have tremendous flexibility. He showed that by measuring the surface tension, he could show that the surface tension of the final stage is what really causes the efficiency of the structures. He's done a lot of work with this, and he showed that, in fact, these things are very, very good at delivering molecules into cell. These are just some of the phospholipids. They're dyed phospholipids. Let's see if I can go back and replay this movie. It doesn't repeat, but you can see they're inside of the cell. And he can show that with a variety of ways that the asymmetry really improves the uptake. It actually improves also the delivery, so it improves the release. In this case, what he did was he put a piece of mRNA that expresses a fluorescent protein, and he shows that you can release the mRNA and cause transfection in the cells, and it's really improved by using the asymmetric structure. Our thinking is that the asymmetric structure looks much more like the asymmetry of the membrane of a cell. And even the cytotoxicity, the number of cells that are killed by the presence of these membranes, always the great concern that you have is, are they safe? The cytotoxicity, the cell killing, is greatly reduced by using these asymmetric structures. Okay, so I've tried to show you a bunch of different structures that we can make. Some of them going back a long time, others of them really exactly what we're working on now. We, of course, think there's many things that we'd like to do with them, but you should now really go back to the question I asked. I'm making these one by one. They're all exactly the same. It's beautiful. It's one by one by one. The problem is, if you make things one by one, can you make things in sufficient quantities that you can make something in a practical sense? So somehow can you scale things up? So is it scalable? And I'm not going to show you all the different things that we've done to improve the scalability, but I'm going to show you just, I want to prove to you that it is scalable. And to me, if it's scalable, I should be able to make something that people will say, this is useful. What's something that's useful? If I can make something that I can sell, that I can commercialize, then it's useful. So can I make something on a useful scale? So remember what I'm doing. I'm really making drops of one fluid another, maybe with shells of one thing. It's essentially an emulsion. So I have to find something that would be useful with emulsions that still people will pay a lot for, because I can't make them that cheaply. Obviously, the thing that I've focused on, I've told you about, is drug delivery. And that's great, but to commercialize something for drug delivery takes a long time because you have to go through a lot of approvals. So we had to find something that didn't take quite as long to commercialize, but we could show that would be something you could produce on a large scale. So what's something that's an emulsion that people pay a lot of money for? Cosmetics. Okay, so can we make cosmetics? So here are these same polymerosome structures, and this is the first thing that we made. These are polymerosome structures that were commercially incorporated into a product made in South Korea, which is really a center for cosmetics. By the way, I learned a lot about cosmetics while I was working on this. But this is very nice, but can we do something even more practical? So here's this company that's called Capsum. This is a production unit. This is actually one of the early production units, commercial device. It made something that looks like this. It made skin caviar liquid lift, La Prairie. How many people know what La Prairie is? Put up your hand if you've heard of La Prairie. Sigrid, you know. Come on, women who know cosmetics should know this, but of course I told you it has to be something with high value added, right? So not everybody knows it. Here's the next page on the website. You can see that for 50 mils is $500. So not everybody can afford it. But this is not even, I mean, this is really a unique product. The Capstone has gone on to make many other products. This is actually an example of a commercial production line, and you can see what it's done. It's basically taken the same capillary tubes and it's just parallelized them. I thought we spent a lot of time trying to make them into chips where we parallelize them. We put a lot of effort into them, but in the end, if you want to parallelize it, the best way of doing it is just make lots of devices and have each one monitored. It takes time to put together this device, but once you've done it, it's all there. And this is what they made. Hydra Beauty Microserum, patented microdroplet technology. Go and look at the website. You'll see it says it's made with microfluidics. And in the first 9 months, this is about 50 mils. In the first 9 months, not running all the time, they made 1 million units of that. In fact, Chanel liked it so much that they just bought the company. Okay, so I've showed you something about making materials. Now let me spend a little bit of time. Anderson went away because he had to go and find space for his new faculty. I'm going to try and save him space by showing you some lab on a chip examples. So I showed you the way we commonly use now, do lots of experiments using robotics. But here, this is a 384 well plate. You might have 100 microliters, typical value, quantity in a well. What we do is we make drops, and we use each drop as a reactor. So now each drop has only a picoliter. Difference between 100 microliters and a picoliter is 8 orders of magnitude. So now we can do reactions with much, 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 much smaller volumes. Of course, to do that, we better really control things, and we better control the way we adjust them. So here's just a simple primer. A 1 micron drop, that's a little bit small for what we work on, but we could. 1 micron drop is a femtoliter. 1 molecule in a femtoliter is nanomolar concentration. Good enzymes work at nanomolar concentrations. You actually know this because the nucleus of a cell is about a micron in diameter, and the idea in the nucleus is to have a very small number of molecules that produce very rapid chemical reactions. It's translating the DNA. So you want to do that, you just do that by keeping the volume very small. 10 microns is a more useful amount. That's a picoliter. 100 microns is a nanoliter. And we do experiments roughly at 1,000 per second. The rate that I showed you before was roughly 1,000 per second. So to do that, the beauty of this is we can do many, many experiments. We use a total volume is very, very small. We can, if you think about this, if I'm using a picoliter and I do 1 million experiments, 1 million experiments, I've only used a microliter of reagent. And really, the cost of reagent is one of the most important costs of any high throughput experiment. But you get also improved signal to noise because you've decreased the volume, so you decrease the noise. The signal to noise, the amount of signal doesn't change, but the noise goes down because the volume goes down. So the signal to noise becomes much larger, and so you increase the sensitivity. So to do this, we better be able to control things. Why is this not working? Sorry, some of the movies don't work. I don't quite understand why. Here's a, let me just go back and stupid thing. So this should be playing, this should be making drops. We can split them into different amounts here. We can split them many times. We can take some out. We're making different types of chips. We make them, we split them. We're controlling them. We're adding here drops. Again, I apologize why this is not working properly. We can add things to make the materials. Here we're injecting a small amount. Here we're injecting large amounts, and well, here we're detecting. I hope this works. No, it's not working. Sorry. I don't know why it's not working. It was working before. So the question is, once we can do this, what are some of the applications that we can do? one of the applications. I'm going to show you one simple application, which we've done now many different times, and we can think a lot of different things to do it. The idea is, can we take an enzyme and can we improve an enzyme? An enzyme is really a very, very good catalyst. Can we make an even better catalyst? So here's an example. This is Amplex Red. It's just, it's horseradish peroxidase. It's a very common enzyme that biologists use as markers all the time. The reason is that it takes a substrate and it makes it into a fluorescent dye so we can detect something. But that's not important. It's just an example of what we can do. So the question is, can we improve on this? Can we do something better? And the way you can do this is not to do chemistry, but to do evolution, to do directed evolution. I don't have her picture in this slide, but Frances Arnold at Caltech won a Nobel Prize about 4 years ago by showing that you could use directed evolution to improve things. So what is directed evolution? It's just doing what Darwin suggests. It's making mutations in the enzyme, in the gene that codes for the enzyme, making enzymes that are different. Most of the time it won't work, but you'll find sometimes it will be improved, and you choose those ones that are improved. So what you do is you take the gene that codes for the enzyme and you replicate it using PCR or some kind of amplification method, and you put lots of errors in it. You put some mistakes in it. Or you take it and you break it up and you shuffle it. Again, these are ways that nature uses to actually evolve something. But nature has many millions of years. We want to do it in a short period of time. So what we do is we create this library, and in the first set of experiments, what we did is we took the enzyme and we put it into yeast. We put it into a plasmid and we put it into a yeast cell. We worked with Dane Woodrup at MIT, who's pioneered this experiments where you display the enzyme on the surface. So you trick the yeast to produce the enzyme, but to have it bound to the surface. And so now it operates things, but you have many copies of it bound to the surface of the yeast. And so then what we do is we encapsulate single yeast cells into the drops, and whenever the enzyme is present, in this case all the yeast are producing the same type, whenever it's present, when the substrate is there, you see that it fluoresces. So we've encapsulated the fluorescence. We don't have to bind the fluorescence to the yeast cell. It's encapsulated in the drop. So now the experiment is actually pretty simple. What you do is you make a library of mutated enzymes. You put them into a yeast cell. You put the yeast cell into a drop, and you just ask, does it operate on the fluorescent? Or does it operate on the substrate? In most cases, because you've put a mutation in it, it doesn't work. So in most cases, you don't see anything. But occasionally you do. And you just look for the ones that you do, and then you sort them out. You take the ones that and you sort them. And I apologize for the movies not working, but one of the devices that we made is something that sorts things. And so if we do this, these are the results that we get. Here's this. If we have only the wild type, this is the average level of fluorescence that we see in all the drops. Not all the drops have cells in them. Not all the cells are working that well, but this is the average level. Then we take a library of mutated yeast, and most of them don't work. Most of them don't work. So now the average level has gone way down. But what we can do is we can sort out those that do work. They're in yeast, so we can just grow the yeast up again. And we can do another sort. And we can do another sort. So we can get better quality of the sorts. And then you can see after each sort, the level of fluorescence goes up until finally, after 3 or 4 rounds of sorting, you get something that's already much more. So you can look at these things and you can see, well, okay, there are now some mutations that seem to work better. And we took a lot of these things, we sequenced a bunch, we sort of figured that we started with something like a library of 10 to the 7, about 10 to the 7 different mutations. We found something like a couple of hundred ones that were better or that would work. We measured how much better they are. Some were not quite, this is the wild type, some were not quite as good, some were a little bit better, a factor of 3 better. Okay, we did something not so bad. But then we did something else. We could sequence them, we could see where the mutations were, but we took the top 20 of those and we mutated them a second time. So we did a second round of mutation, and in this case we immediately found things that worked way better. In fact, they were so much better that we couldn't even measure how much better they were because we were limited by diffusion. So we're much, much better. So what were we really doing? Well, this is a fitness landscape. This tells you how you can think about these things. The higher they are with the, on this peak, the better they are. And nature has made something that works really, really well. And we want to go way over here where there's another peak. The trouble is, as this, every way you move in this direction is some mutation. And most mutations actually take you way down here. So how do you get up here? What we did was we did what was called a neutral selection. Basically, we found a bunch of things that worked roughly as well, maybe not quite as well, and then we did a second round of mutations, and that led us to the top. Now, you might think this is a standard thing to do. The problem is that, remember what I said, we found 100 that worked roughly as well. We started with a library of 10 to the 7. So the likelihood of finding these things was about 1 in 10 to the 5. 1 in 10 to the 5. You had to do 100,000 experiments to find one. By my estimation, in a quantum of time for one of Frances Arnold's students, when she did the experiments that led to her Nobel Prize, quantum of time means a graduate student lifetime, maybe 5 years. That student could screen maybe 1,000 different variants. So if you can only find 1 in 100,000, being able to screen 1,000, it doesn't make sense to do this. But because we can do it with microfluidics, we can do so many, it's easy to do this number of experiments. Actually, I'm sort of lying. You could have done it. Frances Arnold could have done this with a robot and done that. This is the reason why she wouldn't. If you used a robot, it would take about 5,000 liters of volume. 5,000 liters, a liter, 5,000 liters of volume to do the screen. For us, it took 150 microliters. It would have taken about 2 years to do with the robot to screen. The total screening time that we did was about 7 hours. And if you add up the total cost, it would have cost about $16 million. And I calculated this about 10 years ago. So with inflation, it's probably $20 million by now. We figured it cost about $2.50 to do our experiments. We've since moved to much more sophisticated things. Now we don't use yeast. We do things just by doing in vitro expression. We put all the material in to take the enzyme and take the genes and produce the enzymes. And then we don't have to anymore just use one enzyme per drop. Now we can put 1 million enzymes per drop. But the likelihood of finding something that works better is very small. So there won't be that many drops that will have something that works better. So you can see the drops if it works better. You don't know. There may be 1 million different enzymes. Only one of them is working. You don't know what it is. But what you can do is you can sort it and then amplify it and resort it. And so doing this with several rounds of screening, we've been able to screen up to 10 to the 12 variants. Before we were doing 10 to the 7. Now we're up to 10 to the 12. We think we could even go higher with the right conditions. Okay, so again, I apologize for some of these movies not working properly, and I hope I've convinced you that there's a lot of different things that you can do with microfluidics, both make materials from a practical point of view, an interesting science point of view, and do really important high throughput screening for biology. And so with that, let me stop and thank you for your attention.
Thanks, Professor Dave Weitz, for a wonderful talk. And I'm pretty sure we all learned the beauty and power of small droplets by using the magic capillaries. So now is time for the questions. Any questions?
I get to ask you questions if you don't ask.
Yeah. So I saw you produce one droplet at a time. How do you control the size of drop if you want to, say, maybe the diameter of the capillary or any other methods? control?
It's just by controlling the flow rate, typically.
Oh, flow rate.
Yeah, just the relative flow rates of the inner and the outer fluids.
Okay. Can you extend to nanofluidic the similar way?
So the answer is that this is using Navier-Stokes equations. Navier-Stokes equations work down to nanometer scale without any problem. So the answer is everything I showed you can absolutely work at the nanometer scale. The problem is that a piece of dust is about 5 microns in size. And if you're using nanometer size microfluidic channels, a dust will clog the channel. And so you have to be very, very clean. We're just lazy, so we tend not to do that. But there's no question that you could. And so, in fact, what you saw when Kevin, when he wanted to make very sub-micron structures, he used many, many, many channels in parallel. And then a piece of dust doesn't hurt you.
Thanks. Yeah, thanks for a great talk. So I have a question about the interesting the lipid membrane. You make a symmetric membrane. What kind of a solvent and medium you use to mix with the lipid to make this kind of lipsome?
Okay, so actually you're asking something that is still the subject of a lot of optimization. What we really want to do is use solvents that are completely biocompatible. So a lot of the solvents we use are not so biocompatible because we want them to evaporate fairly quickly. And there are a few people in the group and visitors to our group who are trying to understand what the right combination, what the best combination is to make the best structures. And it's looking like you need some combination of solvents, some of which are good solvents, some of which are bad solvents, and the idea is to get the good solvents to evaporate and have the bad solvents still there, because then the lipids will come out of the bad solvents and will form the bilayer structure, but you still want to be able to evaporate it. So we're still investigating exactly what are the best combination.
So for one step, you need to make the emulsion again by using the double liquid, by using the liquid-liquid separation. So for that part, choosing the solvent might be very tricky.
It's not so bad because, again, we haven't worked really hard. We use miscible solvents, but they still form well enough the structures because they're all being snapped off by the outer fluid. So they still form reasonably well, and that seems to work.
Could you elaborate on the directed evolution? I'm confused by how do you pick up the good mutation?
The good mutations are the ones that give you something that works. So you see that the enzyme works on the substrate, and you can tell that because it turns it fluorescent.
Okay.
And so you see the drops are bright. And you just select on fluorescence.
If the if the mutants are not fluorescence, does not respond to fluorescence, so
then you have to. It's always the case with directed evolution. You have to find something that you can detect.
Okay, got it. Thank you.
So you need to have. I mean, so we have a variety of different things. We can do some absorption assays. We can do color assays, but fluorescence is always the easiest.
Thank you.
Okay, because of time issue, and we probably should stop here because later we're gonna have a tea meeting session outside, and if students and audience have more questions, you can have a longer discussion, detailed discussion with Professor David Weitz. Now let's give another round of applause to David Weitz for the wonderful talk. Thank you very much.